Hi, I am Eva Burmer and I will be talking about the process of meiosis 1.
Hi, I'm Rebecca Chen and I will be talking about the process of meiosis 2 later on.
The process of meiosis begins with diploid cells, which are two sets of chromosomes in which you receive each set of chromosomes from your parents. The chromosomes are then duplicated in a phase of the cell cycle called interphase. In interphase, there are three phases that occur. First is G1, which is growth, then is S, which is cell replication, and then is G2, which is growth in preparation for meiosis. In G1, the cells begin to grow in preparation for DNA replication. This phase occurs over the time span of 10 hours. In these next few images, you will be able to see that the chromosomes begin with and end with 23 pairs of chromosomes and 46 chromatids. This is because there is no cell replication here. The cells are just growing in preparation for the cell replication. However, if the cell is not ready for cell replication, it enters a phase called G0, which is also known as the resting phase. The next phase is the S phase, in which DNA synthesis occurs. The cells duplicate every strand of DNA in this phase. During this phase, kinetochores are given to each chromatid as they face in opposite directions and attach to spindle apparatus. For each homologous pair, a sister chromatid attaches its copy at the centromere. S phase takes place in a time span of 9 hours. The final part to interphase is G2. In this phase, the cells continue to grow to make sure that they have all the necessary parts to continue on. G2 takes about 4 hours. In these next few images, you can see what happens during G2. As you can see, it starts off with 92 chromatids. That is because the chromosomes replicated in the S phase. Cells spend a majority of time in interphase. It's important to note that interphase isn't a phase in meiosis, but it's an important phase that occurs before to determine whether a cell is viable to continue dividing. After interphase, prophase 1 occurs. This is the first phase that occurs during meiosis. During prophase, the chromosomes condense and become visible. Once they finish condensing, the chromosomes find their homologous pair of their buddies. This is a procedure that is also called synapsis. They stick together to combine four sister chromatids, also called a tetrad. When the chromosomes find their homologous pair, a process called crossing over takes place. Crossing over allows for different genetic combinations so that every sibling isn't identical. The nuclear envelope also breaks down to allow for the centrosomes to start to move towards the pole of the cells. After this happens, the spindle fiber forms in the centrosoles and attaches the chromosomes at the centrosome. The next phase that occurs is metaphase 1. In this phase, the tetrads begin moving towards the metaphase plate and eventually line up. While the chromosomes are lining up, they confirm that each daughter cell will receive the proper genetic material. Independent assortment allows for the genes to become sorted independently into each daughter cell so it's not affected by genes. Anaphase 1 is up next. The pairs of duplicated chromosomes separate and start to move toward opposite sides of the cell. The cell also elongates and microtubules push poles of the cell further apart. The final step of anaphase is when the duplicated homologous chromosomes are now at the separate ends of the cell. During anaphase 1, segregation occurs. This is when varied parts of chromosomes separate independently. This is also known as independent assortment. After anaphase 1 is telophase 1. This is when chromosomes gather at opposite ends and the nuclear envelope reforms around the chromosome sets, and the chromosomes unravel back into chromatids. After telophase occurs cytokinesis. This occurs along the midline of the cell separated into two new cells. In this phase, the cleavage furrow finally forms to further the separation of the two new daughter cells. Shown in previous images, those are the two halves of sperm or egg cells that will complete splitting at the end of meiosis II.
The process starts over again with prophase 2. The phases are similar, however, there are still few differences between them. The chromosomes condense, and crossing over does not occur during prophase 2. Another difference between prophase 1 and 2 is that the chromosomes do not find their homologous pairs. The nucleus membrane breaks down and the centromes start to move towards the poles of the cell. The meiotic spindles start to form and begins to attach to chromosomes at the centrome. After this, metaphase 2 will begin to occur. During metaphase 2, the chromosomes will be lined up individually along with the metaphase plate. Unlike metaphase 1, in metaphase 2, the chromosomes line up single-filed instead of with pairs. Continuing along, each sister chromatid is attached to a microtubule. Independent assortment also occurs during metaphase 2 to increase gene variation. Then, in anaphase 2, the sister chromatids of each chromosome separate and start to move away from each other. Anaphase 2 is finished off when each sister chromatid, now separate chromosomes, is at separate ends of the cells. The next and final phase of meiosis 2 is telophase 2. The nuclear envelope reforms and the spindle goes away. You can start to see the 4 cells that are going to be left. Then, a final division occurs along the midline of the cell, separating each cell into 2 cells. We are then left with 4 non-identical daughter cells. Now that meiosis is complete, let's talk about oogenesis. In the first division of oogenesis, the cytoplasm does not divide equally. In meiosis 1, the egg cell group will have 2 cells. In meiosis 2, one of the 4 cells will be unequal to the others.